Hi everyone, welcome to my channel. In last session we have seen about the detailed structure of DNA and what is nucleotides and nucleosides. In this session we will see about the composition of nucleosides and nucleotides of DNA and RNA. So let's see what are the names which will be used for nucleosides and nucleotides for different combination of the compounds that is involved in nitrogen bases. So let's see for combination in deoxyribose sugar. What is nucleoside? Nucleoside is the combination of sugar and pentose sugar and nitrogen base. It may be adenine, guanine, thymine and cytosine. So let's see about the deoxyribose, combination of deoxyribose sugar with nitrogen bases. Okay, so first one we will see the nucleosides. So D means deoxyribose sugar, A means adenine, so nitrogen bases, and its name will be deoxyadenosine. And guanine, the formula will be deoxyribose sugar and guanine, and this name will be and this combination of this name will be deoxyguanine. And thymine, the combination of thymine and deoxyribose sugar will be thymidine, the name will be thymidine. And the combination between deoxyribose sugar and cytosine will be deoxycytidine. And nucleotide, let us see about the nucleotide. The formula will be, the nucleotide is nothing but the combination of sugar molecule, that is pentose sugar, that is deoxyribose sugar, and nitrogen bases, and then one thymine, cytosine, anything, and one phosphoric acid, addition of phosphoric acid. So formula will be, so let us see with adenine. So combination of deoxyribose sugar, adenine and phosphate will be deoxyadenylic acid, and the combination of guanine, deoxyribose sugar and phosphoric acid will be deoxyguanilic acid. And a combination of deoxyribose sugar and thymidine and phosphoric acid will be deoxythymidic acid. And combination of deoxyribose sugar and cytidine and phosphoric acid will be deoxycytidylic acid. Okay. So as we see in the nucleosides and nucleotides compounds which is involved in DNA or components of DNA. So let us see the components of RNA nucleosides and nucleotides. So when involved in RNA nucleosides and nucleotides, so uracil will be present instead of thymine. Okay, this is the more important thing. In RNA compounds, uracil must be present instead of thymine. Okay, so let's see the nucleosides compounds. So mixture of, and in RNA it would be ribose sugar. It would be ribose sugar. Okay. So nucleoside formula is for RNA is ribose sugar. Ribose sugar and mixture of ribose sugar and adenine is adenosine. Mixture of ribose sugar and guanine is guanosine. And mixture of ribose and uracil is uridine. And mixture of ribose and cytosine is cytidine. And when we come to a nucleotide, formula will be compound will be sugar, adenine and phosphate. It will be adenylic acid, and a combination of ribose sugar, guanine and phosphate will be guanylic acid, and a combination of ribose, uracil and phosphate will be uridylic acid, and a combination of ribose sugar and cytosine and phosphoric acid will be cytidylic acid. So as we've seen the different names and different combination of compounds involved in nucleotides and nucleosides and its names, let us see the classification of nucleotides. The main reason of seeing these names and this compound mixture of these compounds because of these valuable products. These valuable structures will help for many purpose and many functions in our body. So let's see what is this. These are called as higher nucleotides. This is presence of more than one phosphate group in that compound will serve as an higher nucleotides, and this higher energy bonds, there will be high energy bonds in it. Energy has been stored in their bonds. Okay, so this is the classification of nucleosides. Adenosine nucleotide classification means, so what is adenosine? It's the mix adenine and one phosphate group is known as adenosine nucleotide. So what are adenosine nucleotides means? There are 4 types of adenosine nucleotides, that is, one is ATP, another is ADP and AMP and CAMP. What is ATP? ATP is known as adenosine triphosphate. Adenosine triphosphate, that is known as ATP. So this is the most important compound which serves as an energy compounds in our cells, so which involves in many metabolic process and many DNA replication, bonding, formation and many things which serves as an energy and which gives the energy to work and walking, speaking, talking and many works. This ATP energy is providing us the energy in our body. So this is an ADP and ADP. ADP is adenosine diphosphate, AMP is adenosine monophosphate, and CAMP is cyclic adenosine monophosphate. This is also more important compound which serves as a messenger compound, which serves as a messenger which is involved in hormone transformation, hormone uptake of cells in it. We will see in this detail in later session. So let us know this as a simple basic. So let's know it as simple. So guanos and nucleotides, in which there are 4 types of guanos and nucleotides, that is GTP, GDP, GMP, CGMP, GMP. So what is guanosine? Guanosine is nothing but the mixture of ribose sugar, guanine and one phosphate group. It is known as guanosine nucleotides. So GDP, GTP is nothing but the guanosine triphosphate, GDP is nothing but the guanosine diphosphate, GMP is nothing but the guanosine monophosphate, CGMP is nothing but the cyclic guanosine monophosphate, which is also involved in many calcium forming process. It's also important thing. So this is also a more energy giving compound which is also present in our cells, which also gives energy to our body and cells and many functions. This ATP will be generated in mitochondria in respiration process. So we will see in later on session in detail. So what are the uridine nucleotides? Uridine is nothing but the phosphate group, ribose sugar and uracil. It's known as uridine nucleotides. So UTP means uridine triphosphate, UDP means uridine diphosphate, UMP means uridine monophosphate, UDPG means uridine diphosphate glucose, which is also an important compound which involved in metabolic processes. So it's not important that thing in detail, we will see in another sessions. So this is also involved in metabolic process. So next see about cytidine nucleotides. Cytidine nucleotide is nothing but the phosphoric acid, ribose sugar and one cytidine nitrogen base. So CTP is nothing but the cytidine triphosphate, CDP is nothing but cytidine diphosphate, CDP is nothing but CMP is nothing but the cytidine monophosphate. And also there is some compound is known as deoxycytidine diphosphate derivatives. These are the derivatives of glucose, choline and ethanolamine, which also involves in more important processes. So these are the basic classification of nucleotides. These are more important energy giving products for our cells and many processes involving in our cells and in our body. So this nucleotides plays a very vital role in our daily process of life. The classification of nucleotides and how its important role plays in our daily life and in our body. So let's see about what are the types of DNA. So these are the three types of DNA, that is bDNA, aDNA and zDNA. This bDNA, aDNA are the most abundant DNA which will be mixed or present in our body. So, and I will explain and we will discuss that how this DNA will form, how this DNA has been formed, what are the reason of this forming of this DNA, we will see. So let's see what are the types of DNA, what are these features, what are the characteristics. So these are the helix will be for bDNA will be right-handed, that is this is right-handed rotated, that is clockwise rotated, and this for aDNA also this will be clockwise rotated, and zDNA will be anti-clockwise, that is left-hand direction. So diameter of the DNA will be in nanometer, that is 2.37 nanometer, and for aDNA it will be 2.55 nanometer, and for this zDNA it will be 1.84 nanometer. Number of base pair per turn of the DNA. This is one turn, this another turn, this another turn, this another turn. So per turn of the DNA will be 10, and for aDNA it will be 11, and for zDNA it will be 12. Okay. so it will be 11 for this bit 10. And distance between two consecutive base pairs, that is distance between these two consecutive, this one base pair, this is another base pair, two consecutive base pair will be 0.34 in nanometer. And for ADNA it will be 0.29 nanometer, and for ZDNA it will be 0.37 nanometer. Distance per complete turn, that is one pitch of the DNA, that is complete turn, that is one pitch of the DNA will be 3.4 nanometer, and for ADNA it will be 3.2 nanometer, and for ZDNA it will be 4.5 nanometer. And the major group will be wide and deep in, this is the major group and this is the minor group. This is the major group, that is outward of the DNA, and minor group, this is towards the DNA. So it will be wide and deep, and for ADNA it will be narrow and deep, and for ZDNA it will be flat or absent. There is no major and minor group, it is undifferentiable. So it cannot be differentiated into major group. So, and next one will be minor group. It will be narrow and shallow, that is narrow, and it should be broad. The minor group will be broad and shallow, and for ZDNA the minor group also narrow only and deep. So these are the important features of the types of DNA. So there are another rare types of DNA are present, that is cDNA and ddNA and eDNA. These are the rare types of DNA which is not important, but these are the main basic types of DNA, and what we have seen is that's future only. As we see in the basic types of DNA, so let's see how these types of DNA has been formed. What are the reasons that is involved of forming these types of DNA? So there are three basic conditions that satisfies the forming of these types of DNA. Let's see one by one. So first reason is ionic and hydrated environment. It is the main reason, it is the main environment for the DNA, so which changes the helix of DNA. So ionic and hydrated environment of a cell that helps to change the helix of the DNA. So for aDNA favors low hydrated environment. In low hydrated environment in the cell means the DNA must formed into aDNA. In high salt environment the DNA must be in ZDNA. If a cell contains high salt environment means the DNA should be ZDNA. So it forms into ZDNA. And the next reason will be that DNA forming of the DNA will be altered by favoring the condition by stretching and altering the purines and pyrimidines. The stretching of purines and pyrimidines will ultimately leads to changing the structure of the DNA. So it will change into aDNA or ZDNA. And another important reason is presence of proteins or binding of proteins with one strand of DNA will force the DNA to change into different structure. That it will be, for example, this is the bDNA. That in a cell that there may be any proteins that will attach to the any one strand of the DNA and may change it to the so binding of the proteins of that one strand of the DNA may change the DNA into another's form, that may be aDNA or zDNA. So from this bDNA will be the stable DNA. bDNA it may be converted into aDNA or zDNA with respect to that environment in the cell, and also by stretching of the purines and pyrimidines by any reasons, and the presence of proteins that will binds in the to the DNA of the bDNA and may forms into aDNA or zDNA. These are the three basic reasons which forms the different types of DNA. This is the end of the video. Thanks for watching. Let us see what are the things we have discussed in this session. So in this session we have seen that names of nucleotides, nucleosides and nucleotides, and classification of nucleotides, that is adenosine nucleotides, guanosynucleotides, uridine nucleotides, and cytosine nucleotides. This is the mRNA, RNA compounds, that is uridine, adenosine, like we have seen that the RNA compounds, only classification of nucleotides, RNA compounds, that is ATP, GTP, etc. As we discussed, that ATP and GTP are the more energy yielding compounds which stores the energy in their phosphate bonds involved present in these compounds. So these two compounds, this ATP and GTP, is a more energetic compound. This produces the energy to our body and the cells to do their function correctly. And we have seen the types of DNA. There is BDNA, ADNA, and ZDNA. And there are rare types too, that are cDNA, ddNA, and eDNA. These are the very rare thing which may occur due to any wrong thing will occur in the cell, but this is the more important basic types of DNA. So this DNA, this bDNA and aDNA are more predominant in our, are more abundant in our cells in our body, but bDNA is the more stable DNA, in which by any environmental condition in our cell or any reasons that the condition satisfies, that bDNA will be changed into aDNA or zDNA, but bDNA is the more abundant in our body. So this is the end of the video. Thanks for watching. If you like this video, please like it, and if you want to subscribe my channel, subscribe to the channel. If you have any doubts, please comment in the comment section, and thanks for watching. Thank you.